All right, so just to remind you, you have received the list of seminars for you to sign up. And so please read the instructions carefully, because I did receive emails right away saying, oh, I want to do this one seminar. But the emails, the attachment is very clear. You have to rank the seminars in order of preference, and I will try my best to give you your top 2 preferences. But given that we have a mixture of students, I have to organize it according to everyone's desires. So I will at least give you one of your 2 top preferences. All right. Another really important thing is that a component of this course is participation. So your attendance during the seminars of each other seminars is very important, and I do take record of your attendance. So if you miss a particular seminar, that will affect your the component of the course that has to do with presentations and seminars. Okay, so today what we're going to talk about is a bit about analytical techniques or tools that one could use to assess metals in a biological system. And so I like to talk a bit about this, and it's meant to be an overview, because as you dive into the literature that pertains either to the review article or that pertains to the articles that you select for the seminar, there will be techniques that are referred to, and I would like for you to have some amount of insight as to how this allows you to understand metals, metal coordination, and perhaps metal function and transport. So this is just a flavor for it. Obviously, there is much more that has to be spoken about with regards to analytical techniques that can be used to probe metals in a biological system. All right, so let's go ahead, and I'm going to project. All right, so we're looking at these tools that are available to assess metals in living organisms, and we'll talk about, which techniques are useful in certain scenarios, but which techniques may be more viable for looking at things in a more in vivo context. So as a way of review, in our previous lecture, we talked about these key topics. We explored metals and metal ions in aqueous solution, and we defined formation constants, this concept of the K expression and the beta expression, one talking about a one-step binding process and another talking about a commutative series of bindings. Then we assessed the factors that influence formation constants of metal-based complexes. We explored pH-dependent aqueous speciation and examined its real-life significance. And throughout the course, we will be looking at this much more closely, this idea of how do metal complexes behave in biological fluids. So I want to introduce a term that is closely affiliated with the concept of aqueous speciation or pH-dependent aqueous speciation. It is the metalome. So you probably are familiar with the term omics, which has to do with just a general study of different species in a biological environment, where they're localized, but very importantly, how they display their functionality and with respect to other species in that environment. When we're talking about the metalome, specifically, this was a term coined by R.J.P. Williams, 2001, focuses on the entirety of metal and metalloid species in a biological environment, cell, tissue type, organ. It also accounts for their quantity and localization in the biological environment. So looking at this cartoon representation of a cell, you can see that in all components that make up the omics, right, of the cellular environment, metals are closely affiliated with every feature, right? So I would say the metalome is quite affiliated with the genomics, the proteomics, lipomics, metabolomics, right? So it's something that is pretty much ubiquitous in these areas. So how does one approach the study of metabolomics? So in vitro, there are a number of tools that people use. Methods focus on DNA and protein mutations, so genetic mutations, and studying the impact this may have on the metabome. methods to analyze metal-containing biomolecules, methods to characterize their bioactivity. In vivo, which can be very challenging, there are methods to analyze metal-containing biomolecules and methods to also characterize their bioactivity. So when it comes to really probing a metal species in something that perhaps is coming from a tissue, the question becomes whether to isolate it or not to isolate it. So here I give you a workflow. It's a very simple depiction of how you consider going about studying a metal-containing species, so some component of a metal. And so whether you're working with a cell-based approach or you're working with an animal model, okay, there will be a series of methods that you can use for extraction. And so here's just my, representation of some component that you're trying to extract, let's say some class of biomolecules. Then you have to consider the fractionation that you need to do in order to isolate the molecule or species of interest, then the approach for analysis and identification. And probably as part of this, you might even want to consider tools for to obtain some quantitative information. So when we're considering this workflow and that concept of extraction, the reality is that there is no true one-size-fits-all, because extraction is really a general term. And so whether you're extracting like lipids, nucleic acids, proteins, small molecules, right? There are different methods that you can do so, and depending on if you want something to be soluble, it can be quite tricky to know what would be the right approach. But some things that we need to consider is what are we trying to answer? So let's say that we want to get an adequate in vivo picture, okay? And that, again, is going to be a little bit of a question mark. So, as approximate as adequate as possible. So you may need to use detergents or organic solvents to solubilize molecules of interest. And there are going to be drawbacks with the use of these type of chemicals, as they can denature certain molecules. For instance, proteins are quite sensitive to these things. Chelators like EDTA are used to inhibit metalloproteins, metalloproteases. When people extract blood, they often use EDTA in order to prevent the coagulation, right? But if we're thinking about something that contains a metal, the use of EDTA can be something that is detrimental to the experiment if you want to look at something as it is bound by a specific species. All right, so without inhibition, the presence of active forms of these species may affect the levels of molecules of interest. And then considering the size of what you're trying to extract, so the family of molecules, molecular weight cutoff filters are often used to isolate molecules in a certain size range. All right, so let's consider this simplified perspective of how do you go about the extraction process. So typically what you want to do is collect your sample, and you want to do a process that is known as homogenization. So let's say that you're working with cells, okay? And so in order to homogenize cells, well, you basically have to crack them open. You have to do what is known as lysing them. And the outcome of this would be obtaining what is essentially a crude extract. So here I show you this generalized perspective of the extraction. So you have your cells in a test tube. Typically they will be in a plate. Then you do a homogenization process. Okay. Here I'm showing you a blender. That's not necessarily how it's done, but just to give you an idea that you're breaking it up and you're getting this, what is essentially homogenate. And then often people take advantage of centrifugation, especially ultracentrifugation, in order to isolate certain molecules, but also very importantly, in order to eliminate cellular or organ debris. All right, so then the next thing is considering the fractionation process, the purification process. So here you take advantage of biochemical properties to purify molecule of interest or species of interest. And oftentimes, when you're dealing with something that's coming from living tissue or living cells, right, you're talking about what are known as complex samples, right? So when you're dealing with that, it is very important to consider the use of more than one technique. So to obtain as high a purity as possible, it's essential to consider potentially a two-dimensional or even a multi-dimensional fractionation process. So an approach that all of you are familiar with is column chromatography. So this type of technique takes advantage of differences in molecular size, charge, binding affinity, and other properties. And so there are some typical features of a column chromatography. For instance, you have what is known as a stationary phase. So this will typically consist of porous solid matrix with appropriate chemical properties. Then you have what is known as the mobile phase, which is usually a buffered solution, which will percolate through the stationary phase and is not always constant. And what I mean by that is that the components within the buffer may change, and they may change for a very specific reason to help to isolate molecules of interest. So the complex sample, and I'm talking about the sample that you've isolated, is still going to be very complex. You're going to have a mixture of different things, is layered on top of the column, and the mobile phase causes the molecules to migrate fast or slow depending on their properties through the stationary phase. All right, so that just gives you the generalities of what constitutes a column. And so here's just a cartoon representation of the elution process, where here you have your stationary phase and you load your sample. And here the colors are depicting the movement of different molecules as they are moving through the stationary phase and are separating. Okay, that's what we're seeing here. And then you collect the eluates. Okay, and so you want to be pretty systematic in terms of how you collect. Either you collect in terms of a set amount of time, or you collect in terms of a set amount of volume. That's up to your judgment call. But this helps you to separate and collect these samples. So let's imagine that we want to do a type of chromatography that we're taking advantage of the charge. So to do so, you typically would invoke an ion exchange process. And so here is a technique that's based on molecular charge. So you can either have a cation or anion exchange. And then when doing so, it will now the stationary phase will depend. So the stationary phase will consist of charged groups. But if you're doing cation exchange, these charged groups will be negative. And why is that? Well, they want to capture cationic species that you're going to put through it. And now, if you want to do anion exchange, well, your charge groups will be positive because they want to capture the negative groups that you push through. Okay? And so your choice of cation or anion really depends on the solution conditions. For instance, if you're working in a buffer and the buffer makes your sample positively charged, okay, so then you probably want to do cation exchange. But let's say your buffer makes your sample negatively charged, then you want to do anion exchange. So it really depends on the conditions in which you have your sample, okay, in order to decide which of these will be valuable to you. And you also have to think about, well, what else is in your sample that may help you to improve your fractionation. Okay, so then the mobile phase. So this is what's going to move through, right, the stationary phase, and is going to affect how you obtain your charged species. So initially, your mobile phase is the buffer. And so the idea is that that buffer is priming the stationary phase. But then you load your sample. You, again, pass the buffer through it because you basically want to wash out anything that will not bind to the column. Then what you have to do is you invoke a change in the components of the buffer. You apply what is known as a concentration gradient. And that concentration gradient is essentially of the charged ions that are opposite in charge to the stationary phase. And why is that? Because you want these this component to compete with your molecule of interest that is bound to the stationary phase. So if you're doing cation exchange, well, you want to take advantage of potassium to outcompete the cationic species that are bound to your stationary phase. If you want to do anion exchange, well, you want to use chloride in order to outcompete the negatively charged species. So let's look at an example here. focusing on cation exchange chromatography. And so here, I'm just giving you a representation of different charge species. Okay, so +2, +3, +4, +5. So these are going to bind to a cation exchange column because you have those negatively charged groups that are on your stationary phase. Okay, so then in order to elute them, and elute them in order of higher charge, okay, what you're going to use is a buffer that's going to increase in the concentration of potassium. Okay, so that's what's represented by this feature here, that you're increasing the concentration of the potassium. So that is a gradient that you're applying. And so that's going to help you elute. So in the first part of just passing the buffer without increasing potassium, anything that is negatively charged or anything that is neutral will not bind. But then you will have an elution in order of low to high positive charge. And so that will be the approach that you take to really nicely separate your sample. All right, so that constitutes ion exchange. But another really classical approach is affinity chromatography. So this is a technique based on binding affinity. So here you have a stationary phase with a covalently attached chemical group. And so your molecules of interest will have an affinity for this chemical group. Then your mobile phase will be initially your buffer, because you just want to prime it and you want to get rid of anything in your sample that doesn't stick to the column. But then this buffer will change. You will apply a concentration gradient of an agent with affinity for the chemical group, okay, so that you can outcompete whatever a molecule that's bound to the chemical groups in your stationary phase. Or another approach is to use the chemical group itself, but obviously in its free form, so that you have that competition. So then molecules will elute as the concentration of the agent or the free chemical group increases, outcompeting the affinity constant of the molecule for the stationary phase. All right, let's look at an example. So here, let's imagine that we're interested in the purification of a glucose-binding protein. All right, so that's the objective. We want proteins with glucose that has a glucose component, and so what we want to do is a glucose binding component, and what we want to do is then have in our stationary phase, okay, so these little beads that you see in gray, a covalently attached glucose. All right, so then you put your sample of interest, and so it would consist of proteins, and so you have these yellow molecules there representing a protein that will bind to the glucose. And so in this depiction, in order to elute those proteins that have this glucose binding capacity, well, what we're using is just free glucose, and that free glucose will bind to the protein. That's going to liberate the protein from the beads, okay? And then that will ultimately lead to the elution of the protein of interest. So this is taking advantage of that affinity property. Another popular type of chromatography is one that's based on hydrophobicity and hydrophilicity. So here, so here we're looking at the hydrophobicity and hydrophilicity of the molecules. And in our stationary phase, we can have two different forms. Okay, so we're applying high performance liquid chromatography. And one approach is known as the normal phase. All right, so the normal phase, what we're going to have are the stationary phase consisting of polar groups. Okay, so well, what does that mean then? What that means then, things that are polar in our sample will bind to the stationary phase. Okay, but another approach is to do what is known as reverse phase, where in reverse phase you have nonpolar groups. And so there, things that are nonpolar within our sample will bind to the stationary phase. All right, so it depends on the approach of what you're trying to isolate here. So with that in mind then, the mobile phase will again depend. So if you're doing normal phase, okay? Keeping in mind that the stationary phase will have polar groups, well, then your mobile phase will transition from non-polar groups to polar groups. Okay, why is that? Well, because the non-polar components, well, obviously that's just going to wash through anything that it does not have an affinity for the polar groups of your stationary phase. But then you're going to increase the polarity. And as you increase the polarity, then you're going to elute an order of affinity to the polar groups of the stationary phase. In contrast, the reverse phase will be the opposite, where your solution, your mobile phase, will transition from polar to non-polar. Okay, I will be a little careful here because this idea of polar and non-polar and the mixture of solvents, it's very dependent on the actual experiment itself. Because oftentimes you may have components that would probably traditionally be called, let's say, polar, but because one is more polar than the other, one would be considered the polar component of the mobile phase, the other one would be the non-polar. So that's why I have a quotation marks here, because it's a little bit can be a little bit misleading in terms of that distinction. Really, it's the difference in the polarity of whatever solvent mixer you're using that helps to separate. And so in terms of elution, when we're talking about the normal phase, the least polar will elute first and then the most polar, where in reverse we'll have the opposite. We'll have most polar and then the least polar. And so it really depends on what type of samples that you're working with, which one you think will be a better approach for you. Okay. So again, remember, I'm just giving you a flavor for techniques, and this is not meant to be an in-depth analysis, because a lot of this you guys have seen in your analytical courses, your instrumental method courses. Well, I'm just giving you the perspective that can be tailored towards metal complexes or metal species, and so we'll see that in a moment. All right, so analysis. All right, so let's say you eluted your sample and you have it as, let's say, as pure as possible. Well, NMR can be a really valuable approach, and we're talking about your typical nuclei, your proton, your carbon-13, and then let's say it's for certain metals that may actually be they may actually have an NMR active nuclei, and so here you can take advantage of even solid or solution state NMR. Oftentimes, your sample will not be necessarily 100% pure. What you would have is something that is purer than what it was in the original complex mixture. So it may require the use of two-dimensional approaches. Okay, and so what are limitations? So let's say you're working with complex mixtures coming from biologic samples in probably very precious samples. What I mean by precious is a very small amount of sample. So an approach like this can be difficult to analyze and does consume lots of material. So that is a limitation. That doesn't mean that don't use it, okay? I'm sure people in the lab, for instance, Gemily, I believe, works with NMR. She can tell you how powerful this technique can be. But you have to keep in mind the amount of sample you have, and would a technique like this give you enough information to be able to tell whether you've actually isolated a molecule of interest. Another really important approach is mass spectrometry. So here, solution state study is typically done. There are things that are sort of borderline solution solid state, and I'll talk to you about that in a minute. It usually involves fragmentation of the sample before and or during the experiment. So when we're talking about fragmentation, what does that actually mean? What that means is that you take your sample of interest and then you apply some approach, some ionization approach that actually will break it into pieces. That is what is known as a tandem mass spectrometry experiment. You are looking at the fragments. And so oftentimes you must use a pure standard to verify identification, but not always, okay? And a pure standard, if it's even available, and high mass by charge accuracy. So why mass by charge? Well, because mass spectrometry will typically measure things that are charged. Okay, so you're measuring ions. And so the way it reports the data, it reports in terms of the mass of whatever species is divided by its charge. All right, so another important thing is that if you couple your mass spectrometry to a fractionation process, so a liquid chromatography, like one of these examples that I've just mentioned to you, then oftentimes what people will do, especially if they're working with a pure standard, is they will match what is known as the retention time. What is the retention time? That is the time it takes for the sample to elute. So if that retention time matches the retention time of a pure standard, right, and then you get the identical mass spectrum, then you know you have your sample of interest. But again, keep in mind that you may not always have a pure standard, right? So that is something that we can work around, right? Because mass spectrometry is quite powerful, especially when you couple it to other spectroscopic techniques or other characterization techniques. Now, another really powerful thing is that it tends to use small amounts of material. Now, depending on the amount of material and if you can possibly crystallize, although you don't necessarily need to get single crystals, right? So I probably could expand it. You'll see that I talk a little bit more about X-ray. The use of X-ray crystallography, it will give you information of a solid state form of a compound. It allows for atomic resolution. Again, if you need the single crystals, can be quite difficult, but there are other X-ray approaches that can give you a structural insight and also some elemental characterization. Another thing that people take advantage of, especially when there's a limit of material, is an activity assay. So basically, let's say you're isolating a molecule that has a known property or a known activity. So if you can create an assay, or there is a commercial assay you can use to test whether a fraction that you have collected has that expected activity, that will be an indicator that you have obtained your sample of interest. Okay, so a key thing to note, though, is that the solution in which now you have your sample of interest may not be compatible with the assay conditions. So you might have to consider, how do you transfer your sample into a solution that is assay compatible, right? And then in certain cases, you might even have to think about how those solution conditions may affect the stability of the given species. For instance, I mentioned to you, proteins can become denatured. So you might have to actually consider making the protein now adopt its native form, and that native form may be what facilitates the activity. So there are some limitations here that you have to think about in order to use an activity assay. All right. Before I move on, I want to ask you guys if you have any questions, because we're going to cover, again, just a flavor of different topics, but I want to make sure that you guys have an understanding of this. You can feel free to ask questions always on the chat, and I'll see it. And you can always get into your microphone and go ahead and ask me. Okay, so feel free to ask me as we move along through this. All right. So what I presented to you was not necessarily metal specific. But what if metals are bound to these biomolecules and are necessary for their function, right? So that goes into the question of the isolation, and if that isolation, you really do need the metal for activity, and you're actually, if you are trying to isolate a metal bound to a certain protein or a certain species, right? Well, then how do you go about doing these type of experiments and also analyzing these type of samples? Okay, so an approach is to test the metal binding affinity of the biomolecules. So this would be an approach that would fall under the category of post-isolation. So what do I mean by post-isolation? So what I mean by that is I've done the extraction, I've done the purification, and I've obtained the sample as pure as possible. And so now I want to say, well, does this particular molecule, can it bind a certain metal? Like, can it bind copper, for instance? Well, many people actually take advantage of this type of approach without really thinking about what they're actually doing. So a really popular approach is immobilized metal affinity chromatography, which is IMAC, the abbreviation. All right, so in this technique, you have a chelating agent that is immobilized on the stationary phase. Then a specific metal ion is loaded onto the chelator. And so that specific metal ion would be the metal of interest that you want to examine whether a molecule can bind to it. Then your purified molecule, or even complex mixture, because this could be a form of purification, by the way, is then loaded. And then binding is verified following elution using a free chelator. So the free chelator would be what would compete the chelator bound to the stationary phase to elute whatever was binding the metal. Okay. So note, typically applied for proteins because small molecules would likely overload these columns. All right, so that's a component here you have to think about, is how much samples you have, how complex it is, and would you just mess up your column because it'd just be really hard to purify things. All right, but I mentioned to you that this is a well-used technique, but for reasons that people don't necessarily think about. And so you will often, in a biochemistry lab, take advantage of a nickel NTA column or a nickel chelator column. All right, and why is that? Well, it's because oftentimes, especially when you're working with proteins, you put on a tag on the protein, and that tag is often used for detecting the protein, but very importantly, for isolation, purification of the protein. And so a typical tag is the use of a histidine tag. All right, so histidine. But why histidine? What do you guys think? Why would you want to use a histidine tag protein and then use a nickel NTA column? What do you guys think?
It'll coordinate the histidines.
That's correct. What theory are we taking advantage of here? You guys know this, hard, soft, acid-base theory. That we're just taking advantage of that technique. I don't know, I'm not sure why this happened. This is actually supposed to be 2+. I will fix that if there's actually in my slides. Okay, so, excuse me. So we're taking advantage of the coordinating capacity that the histidine has for the nickel. And how are we taking advantage of that? Well, the NTA that's here, let me clean this up a little bit. Okay, the NTA that we see here, well, that NTA is only saturating 4 of the coordination sites of the nickel. Okay, so I'm not sure why it looks bigger than it actually is. There it goes. All right, so it's coordinating 4 of the coordination sites of the nickel. So you actually have sites where something can coordinate to the nickel, so in this case, a histidine tag. All right, so these 2 coordination sites are available, and we're essentially taking advantage of that. So we're taking advantage of a histidine residue from a histidine tag protein, and oftentimes you tend to use what is known as a 6 histidine tag. What that means is you have 6 histidines in a row to maximize the affinity for the nickel. All right, so that same approach that people typically use for purifying proteins, well, you can use it to try to determine if a molecule of interest, typically a protein, I would say, has a binding capacity to a specific metal. But there would be certain disadvantages. First, it only provides information on the presence of proteins with metal binding sites. It doesn't actually tell you if that occurs in vivo. Okay? Another limitation is that it's ineffective for proteins with deeply buried metal binding sites or that require a metallochaperone, something that will bring the metal to it. If isolated and unpurified proteins retain their physiological metals, well, obviously then their metal binding sites will be saturated and it will be hard to isolate these molecules for whatever metal that's actually on the stationary phase. So this can prove to be very inefficient. Unless you're interested in a specific metal ligand interaction, it probably is not the go-to approach. Okay. Well, what about direct screening of covalently bound metals? So this is something you can do during and post isolation. All right. So in the whole process of the fractionation. All right. So let's talk about the during. You couple an elemental analyzer as a co-detector during biomolecular fractionation. All right, and this is something that is probably not so common, but many people have columns and detectors to look at if a certain molecule is being purified, but they don't necessarily have a detector for determining if a metal is there. But this technique does exist. For instance, high-performance liquid chromatography, which is coupled with inductively coupled plasma mass spectrometry, or ICP-MS. All right, so let's look at the schematic here. Essentially, then, you have your sample. It goes through an aerosol generation process, okay? And then the sample in this aerosol form will then be subjected to this argon plasma that's very hot, and that's going to ionize your sample. So then those ions will then enter the instrument, and then you will essentially discriminate the ions based on their size, okay, or in this case, the mass. And so then you get your detector detecting your ions and then your output of information, okay? So sometimes this can be pretty much straightforward if you're looking at simply an element, right? You can get the trace that corresponds to the presence of that element. The thing about a technique like this, we're talking about argon plasma, we're talking about basically destroying your sample, right? So it is a destructive method. In terms of how valuable it can be in terms of detection, so you can detect elements in some cases as low as 1 part per trillion. All right, let's look at a representation of the data. So here we have the use of 2 detectors, and it may not be so obvious, but you actually have one detector here that is looking at the elution of proteins, and another detector that's telling you whether certain protein fractions contain a metal of interest. So that detector, the ICP-MS, the detector that corresponds to it, is detecting the cadmium. All right, so the traces that we're seeing here. Well, the trace that's in gray, so I'm not sure if you guys can necessarily see that. So I'm going to put that in red, trace over it. Those traces that are in gray, those correspond to a UV vis, or in this case, sorry, a UV detector, and that UV is at 254 nanometers. Okay, so typically when we're talking about 254 nanometers, that actually is a wavelength that's in between 2 important numbers. It's in between 220 nanometers, which will detect the absorbance of the backbone of the protein, so that amide backbone, and also the 280 nanometers. The 280 nanometer will detect the absorbance of the aromatic groups, right? So when we're talking about aromatic groups, we're talking about tryptophan and tyrosine. So people sometimes choose a sweet spot in between those 2 wavelengths of 254, whatever, 250, to be able to detect the protein. So what this is telling you is the fractions in which proteins are eluting, okay? But now we want to see, well, which of those fractions has the cadmium? So the trace that is in black, well, that trace in black corresponds to the cadmium signal, all right? So then here, you can see very clearly that in this sample, fraction number 2 has a protein that has quite a high content of cadmium, all right? And then in the sample below, fraction number 4 has a protein with a high content of cadmium. Right, so that is how you can use 2 detectors to gauge the very same sample while you're purifying it. Okay, so you can use elemental analyzer post-isolation. So that was an example of where you're still isolating it, but here there is a method of post-isolation, although this does not necessarily have to mean that this particular technique you can also use in a way that's not even involving an isolation process. This is known as laser ablation, ICP-MS. All right, so there's a component of a laser involved. Right, so here we have a schematic where you have this laser that is being shined onto your sample, okay, and then that is going to then yield your sample, which will be carried on with the gas. We'll have an ionization process, which would then take it into, excuse me, we'll take it into your instrument. So you have the plasma component that's going to allow for the ionization, okay? And then a detector will detect your ions of interest. All right, so here I'm giving you an example of a sort of post-isolation, okay, although there's still a fractionation of sorts. So we're looking at proteins that have been run through a gel that have been run through SDSPH gel electrophoresis. All right, so here we have a protein gel, and we're looking at simply one column, and so it's not the most well-resolved gel. Okay, so I think Alondra's in the class. She can tell you about purifying proteins with this technique. You want probably a better gel than this, but this is the data that we're looking at. All right, so here you can take the gel and the slices of the columns, or just the whole gel, and then you can put it into the instrument and then shine the laser onto the bands, okay? And then what you're getting is an output of data that corresponds to a certain element. So here, the elements that are being looked at are copper and molybdenum. So apparently, this fraction right here has quite a bit of content of copper and molybdenum. Okay, all right, so that gives you an indication of how you can use this particular technique. Now, as I was mentioning to you, you can also use this technique without having necessarily performed in isolation. And here we're getting sort of an in vivo picture where we're looking at screening of covalently bound metals, or not necessarily covalently bound, but potentially. But we're looking at the profile of different elements in a particular tissue. So here, in this study, a selection of metals and nonmetals from a brain slice, so a thin slice of the brain, was examined by laser ablation ICP-MS. And so we have different profiles that corresponds to iron, zinc, sodium, chloride, phosphorus, carbon. And you can see the distribution of these elements in this slice. All right, so you can get a sense of whether a certain distribution is characteristic of a normal distribution. Now, obviously, this species would have to be dead in order to be able to get the slice, but you're able to use this as a gauge of whether there was a health condition that may be associated with a loss of a proper distribution of a given metal, right? So that, or a given element, that can be quite helpful here, right? So this is I would say, as close to a good snapshot of in vivo, but obviously of an animal that is not living. Okay. All right. So now, if you want to identify metal-bound biomolecules, right? So what approaches are available? Well, there is mass spectrometry. So we're going back to mass spectrometry. Can directly measure the mass spectrum of the intact metal-bound biomolecule. That is, if the binding of the metal survives the purification process. All right, so remember, this idea of extraction can be quite damaging of metal species. So you have to think about the extraction step if you hope to isolate something that will retain the metal. So with regards to mass spectrometry, an effective approach is soft ionization. Soft ionization will help to prevent the loss of the metal, and this is typically achieved by electrospray ionization to detect metal protein complexes, as an example. That's not limited to that. But I'm mentioning proteins as proteins falls into the category of big molecules. But electrospray ionization is limited to pure samples, okay? And then here I will say this is a little bit of, over the years, there's been a lot of improvements. It can give low mass by charge resolution, but depending on the instrument that you use, you can overcome some of this limitation. All right, I see Adriana has a question. There's research on imaging techniques that involve calcium imaging while the animal model is alive, so it's real-time data, exactly. And you will also see, we'll talk about in the course, other techniques that people use while they are injecting a certain molecule into, let's say, a mouse, and they can get a real-time profile of the sample of interest as it's moving through the living species. All right, so that's very much true. Okay, so then here, as I'm mentioning to you about electrospray mass spectrometry, the field has evolved quite a bit. So for optimal and high mass accuracy, you need highly pure protein. That's always going to be the case. also very importantly is not just that the protein is pure, that it is homogeneous. What do I mean by homogeneous? Where proteins are well known to undergo glycosylation, depending on how you've isolated the protein. Okay, if you isolate it from a living thing, or if you recombinantly express it, depending on what you've expressed it from, can be highly glycosylated. So the key thing is to make sure to try to remove all the glycosylation to get your proteins as homogeneous as possible. Now you can get insight at the molecular and even atomic level, such as the binding of ligands and metals and of protein-protein binding. All right, so let's look at an example. And here I'm showing an example of a molecule that we've talked about. So we're talking here about serum transferrin, which we know it's a iron binding protein. So this was a study, 2001. So now this is old, okay? But I'm showing you because I want to show you a little bit of the evolution of this technique. So here we're looking at a molecule that was 40,000, okay? So 40 kilodalton. And so what you're looking at is not the mass, the complete full 40,000 mass, although you are, but in a certain way. You're looking at it from the perspective of one of its charge states, because remember, the data is in terms of mass by charge, okay? So when we see a peak that corresponds to, say, the plus 13, and that's the highest peak, depending on the pH that's being observed, okay? So this was a pH-dependent study, focusing on the 13. Well, basically what that means is that you're looking at something that is 40,000 divided by 13, okay? And then you have to see 40,000 divided by 13. Well, does that make sense in terms of what we're seeing? Something that falls in very close to 3,000. You guys can do the math. You have a calculator there, all right? So you'll see that that makes sense. But what information did they get? Well, they got information on the binding of iron. They also got information on the binding of a synergistic anion. So they were able to get that amount of data. I would say in 2001, they had certain limitations that now they have been able to overcome. So what else are they capable? So this is a paper from 2017. And in 2017, they were able to get a lot of information about different metals that can bind to the protein. So we're still with serum transferrin. And here, I believe we're working with the bilobal protein, because in the other experiment, they were able to construct a monolobal version of the protein. So we're looking at this 80 kilodalton protein, and we're looking at these charge states. So this is a charge state of 21, right? So again, that's 80,000 divided by 21, okay? And so see if that math gives you this number here, okay? But they were able to look at different metals like indium, chromium, titanium binding to this protein, and then apparently several of the ions on. So two of each of these ions binding. What else did they see? Well, they were also able to detect interactions between the serum transferrin protein and the transferrin receptor. So now start thinking about that. How big are we talking about? Well, serum transferrin is 80 kilodalton. The transferrin receptor is 160 kilodalton, right? So now we're talking about a very big protein complex in addition to having metal ions. And they were able to see, again, using the different charge states, the complex with the metal, with the synergistic anion. So they were able to get down to that amount of resolution. It's pretty powerful. But again, it requires the use of a pure and homogeneous sample to be able to see to that level. All right. But now let's say that that you want to look at a small molecule, for instance, containing a metal. And so it's very important using mass spectrometry that you obtain not just the mass of the species, but also another really important feature, which is known as the isotope distribution. Okay, so you need the combination of the mass by charge and also isotope distribution. So here I'm giving you an example of a titanium containing small molecule. Because titanium is an element that has several isotopes of different natural abundance, where titanium-48 is the most abundant of those 5. So when you detect a molecule that has titanium, you're going to actually see those different isotopes. Okay, so here's the representation of that. Let me get rid of. All right, so we have our, sorry about that, I'm going to discard and then start from current. Okay, so now we have our molecule of interest and we see the isotopes, and that will tell you that definitely has titanium in there. If you don't see this distribution, you do not have titanium, even if the mass by charge is correct. Okay, so the MS of the parent ions can sometimes not be sufficient if not compared to a pure standard, but again, having a pure standard is often not readily available. But I do suggest coupling with another technique, for instance, NMR, in order to analyze these type of samples. All right, so here I'm just giving you an example of a titanium compound, right? Because I was just showing you just sort of a generic titanium species. But this is a titanium citrate compound where you have titanium for coordination number 6, and you have this ligand, this citrate ligand, that's binding to the metal in a bidentate fashion, and it's there are 3 equivalents of the ligand bound to the metal. And so what you're going to see is this mass spectrum, and this is a theoretical mass spectrum, because I actually computed this. I didn't actually collect that data. Okay, and there are certain really key features here. So in addition to the isotope distribution that we see here, so this is the isotopic distribution, another really key thing that we see is the charge of the species. So again, remember, this is a technique that depends on ions, and you can detect in the negative or the positive mode. So here I'm doing something that's in the positive ion mode. So what I'm going to see are species that are positively charged. So here I'm just giving you a representation of how I can detect this particular titanium compound as a positively charged species. So you have to form some sort of adduct. Typically you form proton adducts to get a positive charge. And an information that you can get is what is the actual charge of the species that you're looking at. And the way you get that is by looking at the isotopic distribution, and you focus on the separation of the isotopes. Depending on that difference, you can determine the charge. So a difference of 1 indicates that the charge is +1. And I say +1 because we're in positive mode. If that difference was 0.5, then remember, if you're collecting mass by charge, that means that you have a +2 species. If the difference is 0.33, that implies that you have a +3 species. So it's very important how you analyze this information to get the in terms of the mass by charge, isotopic distribution, and the charge of the overall species of interest. That is the power of mass spectrometry. And I mentioned to you this idea of doing what is known as a fragmentation experiment. And that is often done, especially when you have an unknown species. So people use fragmentation to break up a molecule into different pieces, and so they can put the puzzle together. And that can be quite powerful. So in fragmentation mass spectrometry, what you're doing is you're collecting information of the parent ion, and then you're fragmenting the molecule. So let's imagine this is the molecule of interest. You're breaking it up into pieces, and then you're looking at those pieces. So you collect the parent ion and then the mass spectra of the daughter ions to help you put the molecule together to be able to construct what this molecule looks like, to get some information about the structure. And so people can, depending on how powerful your instrument is, you can even do further fragmentations. I would say that you don't want to fragment so much because there may be too much information that you might get confused, right? But it is a very powerful approach. Okay, well, what are limitations? Well, some metals and protein samples and small molecules likely will not survive tandem mass MS fragmentation. Okay, so that's something to think about. Then you may need to couple in vivo experiments with in vitro experiments to verify that an isolated molecule actually does bind a specific metal. Okay, and I would say this is more of a general take-home message, that you often have to do a series of experiments to validate the finding of a metal complex, of a metal biomolecule complex, to verify that species can actually exist within a given organism. All right. So I make this announcement just for those of you who are interested in getting trained on the Molly Toff Toff. I used to do training in person, obviously not anymore. I've evolved this into a virtual training. Actually, it helps me out because I can do this at any time, typically, to as it's convenient for me, but also for you. So if you're interested in applying this technique for your own research, you can feel free to contact me. I won't necessarily do a one-on-one because it's not time efficient, but I will collect a number of people and then we can do a training together. All right. So the last thing that we're going to talk about is identification of metal-bound molecules, looking at some spectroscopic approaches, just mentioning those that you are aware of, because we actually have mentioned a few, but also introducing one that is X-ray dependent. So when we're talking about spectroscopic approaches, we're we're talking about this idea of an excitation process. So when we break down energy, you can break down energy into the translation, rotation, vibration, the electron spin, electron levels, nuclear spin. So all of these different energies that can correspond to a certain type of spectroscopy that can give some really important structural characterization information. All right, so here is your electromagnetic spectrum, right? And then the energy ranges and the techniques that have been built from these. Okay, so at the end, I'll just highlight the ones that you guys are familiar with. But what I want to draw your attention to is to X-ray. So I've already mentioned X-ray in terms of X-ray crystallography, but X-ray can be quite valuable for electronic transitions. So we've talked about electronic transitions, right? For metal complexes, we talked about D to D transitions. We talked about charge transfer transitions. But here for X-ray, what we're looking at is something that's related, but we're focusing on what are known as core electronic transitions, right? As opposed to transitions that can typically be from electrons that are in valence orbitals. All right, so let's focus on that. So X-ray absorption spectroscopy, or XAS. So X-rays have wavelengths that are on the order of atomic dimensions, and therefore these photons can be used to probe the molecular structure of materials. All right, so XAS is a technique that is specific to an atom, right? So keep that in mind, depending on the X-ray photon energy range used, right? So this is going to be dependent on the energy, and let's see why that is the case. So in a typical X-ray absorption spectrum, and I'm going to read this to you, but then I'm going to provide you the visual for you to be able to see this. So in a spectrum, you have a small rise in absorption coefficient, which is observed due to electronic transitions that are coming from the core levels to the valence. Okay, so you're within the atom, okay, a specific element, an atom. You're going from the core electrons that are now excited to the valence electrons, and this corresponds to what is known as the pre-edge. Then you have what is known as a sharp rise in the absorption coefficient, is observed at a well-defined X-ray photon energy called an absorption edge. So what's happening here? Well, now we have the core electrons that are actually dissociating from the atom. Okay, so let's imagine we have this atom, and let's imagine that we're exciting from, and I'm going to draw a really poor representation. So obviously we know we have orbitals, but I'm pretending that we have these orbits, right? You're going from, core orbitals to then valence orbitals. Well, that will be your pre-edge, but then with your edge, you're actually going and you are dissociating them entirely from the atom. Then what happens after that? Well, then above the edge, there are what are known as quasi-periodic modulations. And where are they coming from? Well, they're coming from those electrons that are dissociated from the atom, coming in contact with the atoms that are neighboring to your atom of interest, and then the electron wave produced by your dissociated electrons coming in contact with the electron waves from the other neighboring atoms. And so that's going to produce this quasi-periodic modulation in the X-ray absorption coefficient. This is known as XAS. All right, so the visual, because that's really a lot of words, but not always easy to understand words. All right, so here we have the components of what we were just talking about. That pre-edge, okay, excitation from core electrons to valence orbitals. Then we have the edge, that is corresponds to an experiment called Zane's, And then you have your XFs, the modulation part. Okay, so then let's focus on the X-ray absorption near edge structure, the ZANES. So the X-ray absorption spectrum of a given sample will exhibit an edge at a photon energy equal to the ionization potential of a bound electron in the constituent atom of the sample. All right, that sounds very fancy, but what are we really talking about? Well, we're talking about the energy that corresponds to, exciting, let's say, a 1s electron, a 2s electron, or even the 2p electrons, right? So depending on which core electron you're exciting, then the edge that you're referring to is the following. So the edges are named for the shells of the Bohr atom. For instance, the K edge will correspond to excitations from the shell n equals to 1. The L edge corresponds to excitations from n equals to 2. And then the M edge corresponds to n equals to 3, right? So you're going to have differences in energy corresponds to, where that electron is located. So what is accessible to this experiment? So the accessible X-ray photon energy region is approximately 2 to 30 kilo electron volts. So the lower limit is defined by the sample holder. So basically, what's going to define your limits of what you can detect? Well, your sample holder has some amount of puts some amount of limitations, and so then you have to be able to have an energy that's above 2. Okay, and so the general ones that you're going to see, and this is just a brief list, right? The K edges are observable for these elements. The L edges are observable for the elements here. Okay, so again, there is a way that you can look at a specific energy range and it would correspond to a certain element. All right, so that's a really valuable use of this technique. But why do this experiment? What information do we get? And specifically, we're talking about metals. What are we going to get from this? Well, you're going to get oxidation number. So in general, the higher the oxidation state of the absorbing atom, the higher the energy of the X-ray absorption edge. But why? Right? So remember, it's harder to remove an electron from an atom with a higher positive charge. So obviously, you would need more energy. What else do we get? Well, we get covalence. So it can give us information about the number of paired electrons. We can even get molecular symmetry. What I mean by that is coordination number. So we get some general structural insight. And for some people, that's enough, because let's say you're just interested in determining a metal and its oxidation state and possibly its coordination geometry, then this will be quite valuable. But then there is the extended X-ray absorption fine structure. So here there's more analysis that needs to be done. So the information content of XF's technique arises from the physical interaction of the photo-dissociated core electron with the electron density surrounding neighboring atoms. So that's what I was telling you, that the electron wave that's coming from your electron that's dissociated from the atom of interest is coming in contact with the electron wave from the neighboring atoms. And so this produces this pattern, right? So you have the absorber, your scatterer. So the outgoing photon electron waves propagate to neighboring atoms represented by those open circles. The backscattered waves modify the wave function at the central atom and give rise to the XF's modulations. So that information that is obtained, that can then be processed to give you the identity of the neighboring atoms. And so how is that important? Well, then you can determine what's directly interacting with your atom of interest. You can get some coordination insight. So this gives you sort of a picture of how complex it can be. So here I'm just giving you an overview. We're not going to talk about this in terms of you collect the data and how you process it, and then what form of the data you need to be able to fit in order to determine the coordination of your element of interest. But what's the value, the application? That's what you guys want to know, I'm sure. So XF gives radio distance and helps you answer the following. How many of what type of atom are at what distance from the absorbing atom? So it can reveal the identity of the ligand atoms. But I will note that it is hard to distinguish between oxygen and nitrogen, all right? That can be a little bit limiting because, well, many biomolecules have oxygen and nitrogen, right? So then you have to use your chemical intuition to come up with a model of what's most likely coordinating to your metal, and then looking at the distances and seeing if those bond distances, so that's basically where you're getting bond lengths, are characteristic of a certain atom coordinated to the metal. So So you get some radio structural information within 4 to 5 angstrom radius around the absorbing atom. You get coordination number, right? So you get some insight for that particular species. And this is quite valuable, especially when you're talking about a complex molecule that you can't get a crystal structure for, where you can get some structural information from this technique. But how do you access this technique? So this is not like, doing a UV vis experiment in your chemistry lab or your own labs if you're doing research. You actually have to go to a facility, okay? So national laboratory. So getting access to XF is not necessarily difficult. You just have to apply to get time at a particular laboratory. So you have to write a convincing proposal, typically short, okay? And so you submit it, it gets evaluated, and then you get time. An example of a place is the Brookhaven National Synchrotron Light Source 2. This is a picture of the facility. All right, so what do you need? Well, typically you want to have pure samples. So although there are limitations, but you can work around some of that. You can use in vivo samples. So obviously in vivo is not going to be pure. But if you have a mixture of metal complexes, then it will be hard to deconvolute them. Okay, what I mean by deconvolute them is to know exactly which ones you have. Okay, but you can overcome this limitation by using standards of expected species. All right, well, what do I mean by that? Well, here's an example where someone used Zanes, and they used Zanes to deconvolute the distribution of, to determine a zinc species, right? And so what they did was they collected data for a zinc phosphate, so that the data is right here, a zinc cysteine, a zinc histidine, okay? And then they used that information to deconvolute this experimental spectrum, okay? And so from that deconvolution, they have determined that there was about 50% of that spectrum correspond to zinc phosphate, 25% correspond to zinc cysteine, and 20% correspond to zinc histidine. So that information came from taking a sample, and the sample were leaves. I don't know exactly what leaves they were. We would have to look into that particular reference. But they use standards, and the standards will help them to be able to fit the data, okay? And they have to look into that particular reference. But they use standards, and the standards will help them to be able to fit the data, okay? And they were able to determine roughly how much of these species existed in there. It is a model, right? It does not mean that this is 100% the case, but it is suggestive of what is present in that particular species. Here we see XAFs being performed in a very similar fashion. Here, the person was interested in apparently looking at a nickel species that was in a plant. So what they did was they processed the XF's data to then compare it to the XFs for a nickel citrate standard. And what they observed was that the plant sample and the nickel citrate sample were pretty much spot on. You can see in terms of these would essentially be indicative of the radial distance of neighboring atoms to the atom of interest. Okay. All right, so that shows you how powerful this technique can be. Now, I don't expect you to understand this technique, and obviously we haven't talked about the processing or anything. That's not the point of this. But the objective is for you to be aware that there are really powerful techniques that are available and to your use to determine metal-containing species, right? So for metal-bound proteins, you can also use small molecule standards to predict the metal coordination and the metal binding site. If you have pure sample, then by fitting the XF data, one can determine the primary coordination of metals. Use your chemical intuition or your educative guesses as to amino acids involved coupled with in vitro experiments, right? So you have to couple information to be able to get a final model. This is not something that you give it to the instrument and the instrument tells you this is what it is. No, you have to use your minds to process, to do that critical thinking, critical analysis, and get this output. All right, so this is a summary. So to your collection of known techniques, all right, UV vis, you guys are aware of IR, of NMR. You guys probably have not heard of EPR. Again, that's something that's quite powerful, especially for the metal field, especially when we're talking about paramagnetic species. But to that, you can add XAS, as it can give you really important insight into the coordination of a metal and what's actually coordinating it and its coordination geometry and environment. Okay, so with that, then I conclude my general overview of how you can go about accessing metals, and the hope is that it can help you to think about experiments that are done in the literature that you're going to read and what techniques were used to get insight into the metal species that they're trying to characterize. All right, so if you have any questions, feel free to put them in the chat room or to ask them, okay? But just to remind you guys to read the instructions for the selection of your seminars. I already have some people who have already provided me with their list of preferences. I've given you some time frame to report back to me so that I can to sit down and I can decide how I'm going to distribute the actual seminar selection process. All right then. So if you don't have questions, well then I wish you guys a great weekend, and then on Sunday is Valentine's Day, so happy Valentine's to you guys, and so have a great day. Take care.
Thank you. Have a nice weekend.
Professor, I have a
Yeah.
question about the format of the seminars.
Uh-huh.
We are gonna get assigned to groups depending on like who presents that day, but we select our own papers and everything.
That's correct, yes.
So, but it's that I read in the instructions that we have to do, or it would be ideal to do like a collective introduction and like linkers between each article. Like the articles are going to be
I said
presented the
to give
same
us an
day.
overview of what we're going to learn in your presentation so that we have a nice preview so that when you transition into your respective talks, there is a sense of coherency.
That right. So like all the articles from that specific topic are going to be that same day.
For the session in which a particular seminar will be held, yes.
Okay. So it's just like one introduction for that day.
Right.
Okay. Thank you.
You're welcome.